Hi, today's tip of the week is a really quick introduction to a nice tool we just found that allows you to visualize genome evolution, synteny, and gene duplication. The tool is called Genomicus, and it's found at this website you see right here. It will be linked in our blog post. And basically, it's very simple and straightforward, but has lots of information. So let's do a quick gene search. We'll type in per. You'll see there's 164 results of a gene that starts with the term per. These are, when you search, either ensemble nomenclature or approved gene symbol. We are then going to choose a human genome. You'll see there's some suggested genes. I'm going to do per1. We'll click that. You'll see immediately our reference gene with the heavy blue lines, the phylogenetic sequence to that. And we'll see our human there. You'll also see ancestral genome gene information here in the italic green and the extant species genes in the blue. We are going to make this a little bit simpler to view. Scrolling over and go to the tree menu, go down and hide all ancestors. So we're going to hide those species. We just want to look at extant species. The other thing I'm going to do to make this a little bit easier, you'll see some things here. These are only partially sequenced genomes with a part of a scaffold. We want only those species that have fully sequenced genomes. So we're going to remove all 2x species. So we'll get rid of that. And as you see here now, we have dark blue squares. We have red squares. The red squares is a duplicate of a gene, and these are the ortholog branches. You'll see the little dots here. These are the branches that we hid when we hid the 2x species. And we can click on that to show that node if we want to, in this case gorilla. We can also look at the duplicate. So if we take this and we scroll down here, we'll see the duplicated gene here in other species. This is the per gene, and if we scroll down, and we'll notice that it is the per2. If I scroll over a gene, it will be highlighted, the orthologs in other species, as you see here. So I can scroll over any of these, and it will give me the ortholog and the other genes. Here's an interesting one. If I scroll over there, you'll see VAMP2 has orthologs and other genes, but you will also see that VAMP2 has the ortholog is also a neighbor of some of the paralogs of per1 or per2 in this case. You'll also see later in evolutionary history there were also duplications of per2. For example, in zebrafinch, a recent duplication. We can also change the view to get a default tree topology. So I'm going to go from this topology to one that I actually find a little bit easier for me to view. And now the reference species, the line to the reference species through the branches will be in dark blue, as you see here. It won't be the top species. This topology will remain no matter what you choose as your reference species. You can change the reference species. So for example, let's change it to horse. Just click horse, and you'll see now that my dark blue line goes to horse, gives me a view, and the orthologs in other species in reference to the horse genome. So if I went and let's click fugu. So fugu is now my reference species, as you see here, and now it shows the orthologs in reference to fugu. There's lots of other things you can do here. If I click on any of these genes, for example, it will give me a gene details page. It will give me links to PubMed or Ensemble, UCSC in some cases. I can switch here to that gene as reference if I want to, and it will give me a reference, that gene as a reference, etc. Let's go back. There's a lot more to do here, and so in our blog post I will link you to a longer introduction from their page and a reference. It's a great tool. Check it out. It gives you some great visualization of genome evolution, gene duplication, and synteny. Thanks, and we'll see you next week.